Okay, so we are recording and we're in chapter 11. So I really like how the order of our chapter 10 and 11. Again, as I've mentioned to you before, this is just a personal opinion. I think chapter 10 is could be the most important, not relevant, important chapter of the entire book. And the reason I say that is because now that you're looking into the cancer stats, now that you're understanding the epidemiological data, you hear that word around, right? That word epidemiology. You hear it when you have Dr. Azar, I believe his name, Secretary of Health, when he talks about epidemiologist or epidemiology, or you hear Dr. Fauci or another of the other national doctors and CDC, when you hear that word epidemiology, now you are understanding what that word is and what does it mean. So it's gathering data to try to draw some conclusions in terms of human health. It could be bacterial, it could be disease, it could be whatever, right? It could be autism, whatever it may be. It is the gathering of information used by those in the human health field. So epidemiologists do epidemiology. So I do think 10 is the most important. And that's just, and it's okay if you're like, hey, I think chapter 1, experimental design, is important. Hey, awesome, cool, no problem, no arguments here. It's just opinions. What I'm saying is definitely not a fact. Uh, Please do not confuse this with mitosis, right? No T there. It's not mitosis. So we're going to look, and of course, every chapter starts out with a little picture, but you're not too sure what's going on with the picture unless maybe you've read the chapter, your e-text or your actual textbook. But it shows something. And not, I mean, and I think every picture they put in the chapter is pretty good. So far, so good. Here, they're showing something very interesting. And you can see that there's this shirt and there's this lungs. So the authors of this chapter may be indicating that, hey, I don't know, we're going to talk about lungs. Is there something breathing going on here? What's going on with this situation? Because the background's a little blurred, right? A little blurred. So we're going to assume that there's not much significance in the background. And I really like you to look at pictures like this. You look at the grass, it's pretty even, nice grass. I don't quite see anything going on here that's of real importance. To me, I'm assuming it's involving this person. And look at the shirt, something's going on there. How does the organization of chromosomes, genes, and their alleles contribute to human traits? And how does meiosis produce genetically diverse gametes? And we'll talk about those gametes. I've briefly discussed these before. Why do different traits have different patterns of inheritance? And what do some practical applications of understanding the genetic basis of human disease? So practical applications of understanding the genetic basis of human disease. And this is where I really try to focus is more a little bit more on point 4 because that's where we get to apply some of our learning throughout the chapter, just like we did in our cancer chapter. And then I added more information for you. So you've probably heard of this before, cystic fibrosis. This is a rough one, ladies and gentlemen. This is a rough one. I'll follow the PowerPoint and then I'll add as I go through. This is a genetic disease because look, right? Look at the chapter, simple inheritance. So you're inheriting something. It's not, some people say, I had an inheritance, right? A nice, someone left you a nice property or a lot or a home, or well, hopefully you didn't inherit debt by someone not paying their taxes or something, but hopefully you get something good, bad. Well, in this case, it's not good, it's bad. And cystic fibrosis, so genetic disease. Now, it is caused by a single gene mutation. Remember, ladies and gentlemen, we talked about in chapter 10, in cancer, we talked about what is the common denominator, regardless if it was from smoking, whether it was from the sun, whether it was from chemical X, chemical Y, chemical Z, doesn't matter. Whether it was the rubber ducky that had paint on it that caused cancer, whatever it was. There's a book, by the way, I believe it's called, what is the book called? I'm sorry, you know me, my mind, I'm like a little kid. I can't help it sometimes. If you get the chance, this is a nice read. It's a nice read. Doesn't get too in depth, but it's an interesting read to get you kind of start thinking about toxicology. It's not for the toxicologist like myself, but it does a fair job for communicating information for a person who does not have that much background. So, slow death by a rubber ducky, how the toxic chemistry of everyday life affects our health. Good read if you ever get the chance for that. So that's where the little rubber ducky went in my head. It was like, oh, and that's why I said rubber ducky. Just so you know, that's my crazy little mind. All right. Let me close that. We go back to the PowerPoint. So I was saying that regardless of how you want to slice it or dice it, the common denominator when we were talking about cancer is a change in the nucleotide sequence. And then you're going to say, wait. I got that. I get you. We covered that in chapter 10. So no matter how you want, again, slice it or dice it, when it came to cancer, it was a nucleotide sequence. It's a change in the nucleotide sequence. Okay, got it. And then that caused, right, a change in central dogma. We go from DNA to RNA to protein, and then we saw how certain proteins can be altered, and then they lead to certain things causing, like your p53 tumor suppressor gene, right? Got it. Well, wait a minute. We're not in the cancer chapter anymore. So why are we, what is going on here? Well, not all mutations lead to cancer. It is a gene mutation. Yes, it is a nucleotide sequence change, but not all lead to a mutation. So, what does it do? What does this gene mutation do? Remember your central dogma, ladies and gentlemen. Again, what is central dogma? It goes from what to what to what? From DNA to
RNA.
RNA, from RNA to
RNA. Oh. ATP.
Is it protein?
protein. Yes,
Protein. There you go. Sorry.
protein. So let's say it again. So I'm going to say the word and then everybody say it after me. All right, everybody unmute. Okay. The central dogma goes from DNA
DNA.
to RNA
RNA.
to protein.
Protein.
Good job, because I'm going to ask you that. That's on your exams. You have to, and I keep reinforcing this, guys, because that is the truly one of the, it's like the pathway of life in biology. We have to go from DNA to RNA to proteins. Now, of course, with the exception, as I've told you before, with viruses, right? It's a little different there, a little different, but even bacteria follow that pathway, okay? But viruses, they're different. We won't go into virology, but as I explained the whole thing, how most viruses do not follow the laws of the living. They're in that little weird realm in themselves, right? Where they lack the ability, their reproductive machinery, which is why they hack our cells and they overtake that so they can replicate themselves, usually at the result of death or damage to the host. Don't ask me the why they do it. Why has this happened? It's been happening since before we were here. I don't know, but it's been happening for a very, very long time, the virus. I've always wondered what was the first virus? Like our story on fire, like how did we get fire? I've always wondered what was the first virus and how it came about. And I don't even have an idea. So, central dogma, DNA, RNA, or protein, we have a gene mutation, a shift or a change in the nucleotide sequence resulting in a problem with a protein. So what we have here is we have an allele for the CFTR. What's this mean again? I think it's called cystic fibrosis transmembrane regulatory gene. Don't quote me, but I think that's what it stands for. It'll say it somewhere along in the presentation, somewhere. Does it say here? Yep, CFTR, the cystic fibrosis transmembrane regulator gene. Okay, so the CFTR gene codes transmembrane regulator protein. Ha ha ha! Look, see that? You see that? How the CFTR gene codes for a transmembrane regulator protein, right? Remember when we talked about that P53 tumor suppressor gene, right? And if that gene is off, then you don't have that thing to help you suppress cancer problems, right? Okay, here we go again, but except we're not talking about cancer now. Again, we're still talking about a regulator protein, hence central dogma, DNA, RNA, to protein. So we have a protein problem. We have a protein problem, Houston. We have a protein problem. So let's read this little piece here. So cystic fibrosis is caused by a variety of mutations in the cystic fibrosis transmembrane regulator gene that sits on chromosome 7, right here. One such mutation consists of a deletion of 3 consecutive nucleotides. That is where that's where the problem starts, which then creates a CF-associated mutant allele. Tell me more. Consequently, the protein, central dogma, DNA, RNA, protein expressed from this allele is missing an amino acid. Because why? Because all proteins are made of amino acids. And the ground goes wild, which renders the protein non-functional. Why non-functional? Because structure leads to function. That's right. Alter the structure, you alter the function. So here we go. Nice looking CFTR gene. This beautiful, nice little codes of A's, T's, and C's and G's. And then from central dogma, from DNA to RNA to proteins, and we have a nice functional CFTR protein. Look at that beautiful protein. By the way, please, for the record, this is not what it looks like. This is not what it looks like, okay? Let's do a pause here because I don't want you to get confused with this. That is not what these things look like. That is just to help us put little cartoon images in our heads to figure out what that looks like. What we really have to do is we have to go to X-ray crystallography. That is right, X-ray crystallography. So what can we do? We need to look at let's go. Look up something. Probably some images to kind of find something. Yeah, this one looks good. This one looks good. Let's take a look, see at what we have here. Okie dokie. Maybe I'll bring my friend Ashish Sachan. He did a lot of X-ray crystallography practice there in the Department of Energy. The United States Department of Energy. So let's go to share. How do we know what the heck these proteins look like? How do we know? Well, I'm going to show you because I don't want you to think that, where did it go? Okay. I won't go into all the concepts right now because we don't have the time, but I need you to understand when we talk about the central dogma, which goes from DNA from DNA to RNA to protein, the only way we know what the protein looks like is we have to crystallize the protein. We must turn it into the crystal. So we can then have X-ray diffraction pattern, get this baby here, do a super fancy dance of mathematics, and then boom, create an electron density map, which is then boom, is going to create your protein model. This is what the proteins really look like. They do not look like anything that your book shows you. Nothing like it, right? They're just trying to show something for you to kind of be like, oh yeah, there's a change. I see it, you know, but just have to throw it out there. I don't want you getting confused and thinking that, oh, guess what I saw today, Mom? I saw a protein. You didn't see a protein. You did not see a protein. Now you saw a protein because I showed you what the protein looks like. So or one of you may be talking to a 1-year-old, 2-year-old about cystic fibrosis, you know, and I just don't want you giving that poor little child incorrect information on protein structures, okay? So must be accurate at all times. So we have this little, this picture here, which is trying to depict something, right? So it's trying to say, this is pretend. It's a pretend picture and says, okay, look, this is what cystic fibrosis looks like, right? A nice, not cystic fibrosis, but the gene that goes for the cystic fibrosis transregulator, transmembrane regulatory protein. And yes, it looks really nice. Okay, fine, great. Now, what happens if there is a problem and 3 nucleotides are deleted? Oh boy, the structure changes. What does that mean now? Structure changes function. What is the function going to change? I don't know. Let's see. Why did I stop share? I have no idea. Please forgive me. I left for a few days and it's like I don't even know how to navigate Zoom anymore. Okay, let's see. I'm gonna skip this part. I'm gonna come back. Because I want to go from structure to function. Oh boy, we're really gonna. Okay, here we go. Now everything else we're gonna cover, but I showed you that protein, the normal one, and then the one that has some problems going on with the nucleotide sequence. And we talk about structure as function. So what is the function? What are we doing? What are we talking about here? This is why I jumped all the way over here. If your CFTR protein is not in the correct structure, the following could happen, and it depends. We will then go back and to see there are different types in terms of, we'll get into it, whether you can have a different severities of cystic fibrosis, okay? Different severities due to the genetic mutations. Did you get it from just mom? Then you may be able to deal with it and maybe get through it. Did you get it maybe from mom and dad? Oh, then that's a problem. There's probably, unless you get a lung transplant, it's endgame. And we'll go into it a little bit later. So, structures, function. The CFTR membrane protein facilitates the movement of the chloride ions when the CFTR protein is not working. And why is it not working, guys?
Because there's a change in the nucleotide gene sequence.
Yep. Which then causes the protein structure to
Distorted. Or
change, distort. You're right. They're all the same. Tomato, tomato, potato, potato. Correct. And structure leads to
Function.
oh, hallelujah. Oh boy. My goodness. Thank you so much. Good job. All of you are now like qualified. I will give you an honorary PhD now, guys. Good job. You are understanding the true foundations of biology and disease. Good job. Let's get into a little more details here, shall we? Well, it's not working. And exactly what is it's not working? So what is it doing? Well, chloride ions are trapped in the cell. Okay. Since the chloride ions are trapped in the cell, this results in a thick, sticky mucus on the surface of the cells that traps bacteria, leading to infections. Now, it is very important to know that wherever there is mucus, mucus love. It's like the little spa of bacteria. They love mucus. They love to hang out together and divide and conquer and cause problems. That's why it's very important to also keep them sinuses nice and clean. If you got some allergies, which then can lead to post-nasal drip or some sort of stuff like that, or maybe a little drainage in the back of the throat, you can have throat infections. It's good to have gargle, maybe create a saline solution, look it up. You can figure out all the guidelines to that and how much to put in this and how much more water. If you're going to then do nasal rinses, please use deionized water for this to be on the safe side. And that can help a lot. I do it, and I don't even have nasal problems, but I do it at least twice a week to just keep that nice and clean. Because if I don't do that, I could since I do a lot of talking, I could sound like, and right now we have a normal lung to the left side here, and we'll look to the right. You can clearly see that we have there's stuff there. So it's very important to try to have some good flow going on. Now, let's talk about the normal lung and then the lung that's not so good, right? In cells lining the lungs, and the reason I'm reading it because I've read it before and I approve. I like it. I like the way it flows. I think it's pretty good. If I don't like it, I don't read it. So the CFTR membrane protein allows passage of chloride ions, right? They're passing through the membrane, right? Now, when ions can leave the cell, water is able to flow back across the membrane freely. So it's like it's a two-lane highway. When the ions are not flowing, you don't have that happening. So let's repeat. When the ions can leave the cell, you have water flowing. So let's take a look at that. You see these chloride ions? We have them here, right? And what we're looking at is, it says the functional CFTR protein. So remember, we have a protein And remember, from chapter 3, we talked about proteins, and this is a protein embedded in the membrane, right, of these lungs. And what does this protein do? It transports these chloride ions out. If this little protein, the CFTR protein, is altered and the structure is altered, then these guys begin to accumulate. And you do not have the passage of water. Now remember, water is coming across through here, not through the protein, but through osmosis, right? Water flows freely in both directions across the membrane. Hydrated mucus is thin and slippery. It's nice and hydrated, doesn't make a big, like not a bunch of accumulation of it, right? But now we come over here. And what happens here? Look at those proteins. Because of the mutations in the nucleotide and the pathway of the central dogma, we have an RNA that coded for the incorrect amino acids, which then resulted in the incorrect protein structure, which then leads to a dysfunctional, distorted, improper, non-functional, whatever you want to say, protein that cannot get these darn chlorides out. So they accumulate. Now the concentration changes, right? Water flows into the cells to dilute the high ion concentration. So because now, hey, lots of chlorine ions here, I need to get in here and try to dilute this stuff. Well, what happens is the dehydrated mucus is thick, trapping bacteria and inhaled particles. So it doesn't do a good job here. So we take a look at this one, we'll read this one. In the case of cystic fibrosis lung, the incorrect or mutated, it should say that, the mutated, improper, distorted, whatever you want to call it, right? CFTR protein is not working in these cells, disrupting chloride ions flow. The cells retain more water, causing the mucus outside the cell to become thicker and hard to clear out of the lungs. Bacteria is trapped in these mucus, remain in the lungs, and cause infection. That is the story behind this. Right here. What I just told you is the result of this and this. So then you may be asking yourself, okay, wow, we went from that nucleotide mutation to all this over here. Yes, we did. And now you may ask yourself, I'm going to have to go one step further because I'm following the causal chain. Then we're going to come back. I think that's the way it flows better in the student's mind is now the students is like, what happens now with the lungs, Dr. Delgado? What happens? Okay, well, it depends. There are multiple scenarios that can occur. The good one is where someone doesn't have it that bad, and we will get into the genetics of that and the inheritance of it. And there are some antibiotics and some therapy, and you see that lady and she's running, so I'm assuming she has cystic fibrosis, but she's able to run. Now, there are other cases where it's not that good. That means some of her CFTR proteins function. There are other situations where people, all of their cystic fibrosis transmembrane regulatory proteins are incorrectly shaped, leading to improper function. In that case, these individuals have to take antibiotics quite a bit every day of their lives. And I think like all of you know that if you take medication over and over and over, it'll lead to other issues. can you imagine taking antibiotics that much every day and it's killing off all the other good bacteria? They're going to have some problems with the digestive system, to mention a few. Then they could go to therapy where they invert them. They can't really cough well, and they like pat their backs. There is also a little compression vest, and there is oxygen. They pretty much have to stay on oxygen. They can't run. They can't do many things, and usually results in death if they don't live too long. So those are the outcomes. Now there are some videos I've shown before. I haven't shown them because a lot of students like they get, I mean, and myself too. You know, there's this one girl, she's a, what do you call it when you're popular on YouTube? A YouTubist? Or what is it? YouTuber? When you're
A
popular?
YouTuber?
Oh,
Or
what?
like verified?
Yeah,
Whatever it's
definitely
called.
verified, but what is it like when people know you on YouTube? What's it
I
called?
just hear YouTube.
An influencer?
I don't
Something
know.
like that. Yeah, yeah. She had
Yeah.
cystic fibrosis, and she shared her struggles and everything, and I shared some of that with students, and man, it was a tearjerker, you know. And I just thought this semester I'm not going to do it. I just take a break, just take a break from it. It just every time I share it, it's rough. And young girl. So, and she's talking to people like, not gonna be here, not gonna make it, you know, her good days, her bad days. And to hear someone know that they're going to die is like the hourglass is losing sand very quickly. It helps us, those who do not have that type of problem or very severe health problems, put things in a little bit of perspective. So, but this time I've decided this semester I'm just gonna hold off on it. If you'd like, I'm sure you can just YouTube it. I'm sure she will pop up. She had many subscribers. But yeah, she influenced a lot of lives and research and so forth in that. Don't quote me, but I think she did have a lung transplant, and then I think later things went wrong with the transplant. So, um, so those are some of the things that can happen. Let me go back to the share, and let me see if I don't remember this too well. If I can get to the part where we're going to come on this meiosis and all this genetic stuff, but first I want to. No, they don't. They don't give a lot of it. They don't talk about a lot of it. Hmm. So let's take a look at this chart, right? And at the least, so you can kind of grasp what I'm saying. It says here, cystic fibrosis life expectancy. So as a result of medical research and the development of effective treatments, the life expectancy of people living with cystic fibrosis has increased by 10 years in the last 2 decades and doubled since Emily's diagnosis. I believe this is the individual that I was referring to. Could be, may not. I don't know. You know, I'm really bad with names. But look where it used to start. You know, used to the average person, it says median. The median predicted survival 28. At 28 years old, end game. You see 32, it ends there. Now it looks like, hey, little kind of approaching 40, you know. And of course, remember these things here. Remember these little lines, ladies and gentlemen, because I could put this up there and ask you this. This is called standard deviation. So write that down, the standard deviation right here. That means that, let's say this number right here, we go all the way across is 38. Okay. that some individuals fall close to the 40. That's like the most, and of this data set, some of them were close to the 37. That was the overall range of the lowest of the group, 37. The most that lived the most was, let's say, 40. But the median, not the mode, but the median, it's not an average. The reason it's like an average, but it's not an average because the average is susceptible to the outlier. Remember that. Outlier. Remember we gave that example, the students in Crystal Springs High School with the person who's standing in the back who's 6'3". Then I show up to class. I'm like, man, a lot of people are going to be tall in the Crystal Springs biology class. And that person, 6'5". And I show up like, man, I did an average. And like everyone in the Crystal class is like, I don't know, 6, like they're like 5'8". I show up, I'm like, what the heck? Like, what? No, everybody's not 5'8". The average class is not average height, not 5'8", because that dude in the back was super, super tall, and his data threw everybody's height up, right? Or you have someone who's really short, and the same thing, you could throw the height all the way down. So remember that outlier, right? So this is a median, which is the median is immune to the outlier. So that's what we're looking at here, right? I'll probably have to show you some more. I'm going to show you some more information on some of the health replication, help therapies is what I'm trying to say, help therapies. All right, so now that we understand, and this we may not be able to finish this one in the second we might have a part 3 to this one. Because I don't want to push this content. It's pretty important, and I know this is going to be all new to you. But now that we have an understanding, right? We know central dogma, with DNA, RNA, protein. We have. This whole point of this presentation was to build the setting for you, build the structure for you to stand on. So you now know, okay, in this chapter, I know what the central dogma is. I know that there was a problem in the nucleotide sequence. It coded for an incorrect RNA, which coded for incorrect protein because the amino acid sequence got changed up. Amino acids make the protein structure then changed, which then resulted in a non-working or ineffective cystic fibrosis transmembrane regulator gene, which then didn't allow the chlorines to go out, which then resulted in a lot of the water to go in, which then resulted in the accumulation of mucus, right, on the lung tissue, which then resulted in bacteria having a freaking, a rave going on, and up there, and all that mucus going on, they're just having a good old time up there, which then results in infection, which then results in respiratory problems, which then results in lots of medications, which then could have side effects, and it's just what we call a domino effect, and not in a good way. So, and then you saw life expectancy with researchers going up, but still 40. That's it. Like, yeah, it's better than 28. I'll give you that. It's doubled. But, and this is where I was telling you about, if you can change in the beginning between sperm and egg, when I told you that sounds like sci-fi fiction stuff, right? Or you can then change and help remove the error remove the mutant gene and fix it before the individual is born. That is some crazy stuff, but mark my words. We thought a lot of things were crazy, and we see a lot of things have happened that we thought were all sci-fi. So we will see where we go from there. What I want to do is make sure, before I do this part, because this part can get a little I'm going to go through these slides briefly. I want you to take a look at them. Let me click it. Come on, you can do it. We're going to cover things like genotype, phenotype, diploid. We're going to cover haploid, sexual reproduction. Okay. We're going to talk about this. Making babies, how that happened. Not the process, ladies and gentlemen, like more of the from the egg and sperm, how that works. Okay. Just making it very clear. We're talking about sexual reproduction on a scientific level. Gametes, meiosis, zygotes, embryos. We're going to go through this phase. I call it M1, M2. We're going to take a look at this here through this process, not to be confused, right, with mitosis. Genetic diversity, recombination independent assortments, independence assortments. And then we hit this part and inheriting cystic fibrosis, thinking of it as it being recessive or dominant. Major differences here. Whether are you going to have a little a or a big A? If you're going to have this horrible disease, trust me, you want the little a. You don't want the big A, because that is not good. As I told you, we're talking about here. These are the differences. So we'll get into this. We'll get into a little bit of the Punnett, not so much Punnett square, but we'll briefly discuss it. Maybe I misspoke. Maybe it was recessive. Maybe it was little a. Sorry. So we're gonna have to take a look into this type of this, and this is why I'm saying it might take us a part 3. Okay. Now, in order to make this process work for you, those who are here, I need you to take a look. If you haven't been reading your chapters, you need to read this. Remember, it's for your final, right? So you want to go through at least the PowerPoints, at the very least, your PowerPoints. What's recommended? Going to the PowerPoint, reading your chapter 11. Because the chapter 10, you got it in the bag. I know we drilled and you have it. This one here, now we're getting into some other complicated different things that I don't think most of you talk about on a daily basis with your friends or loved ones. It's just not it's not normal conversation. So I'll pause it here at 11:05, and I just want to make sure that you take some time to go through that now that we have a very, very good foundation. Okay, so remember, Wednesday we'll talk about some cancer stats. We're going to go in between the cancer stats. If I run out of candy, I will then jump into the chapter 10, right? Remember the parade analogy, throwing the candy. And then do that. Open up chapter 11. Do a quick little Q&A before we start. Central dogma. What are we talking about? Cystic fibrosis, proteins, amino acids make proteins. We're going to go into structure, function. Make sure we understand that the chlorine ion, that the transmembrane regulator protein is not transporting the chlorine ions. Get the foundation knocked down, and then we're going to jump into the other parts of that presentation, and then we'll try to finish it. But we might have a part 3. This will be probably the only presentation we might have a part 3, because I don't want to just speed through it. So just letting you know that. Okay, I will stop recording now.